Good, go on. Once upon a time, there was some tissue maintaining homeostasis. All of a sudden, a splinter broke through the skin's protective barrier and it's covered in pathogen. The mast cell recognizes damage and sends out histamine. The histamine goes to the blood vessels, which causes it to become dilated and leaky. The neutrophils come into the damaged site and engulf and destroy pathogens. The macrophage also engulfs and destroys pathogen, and this macrophage becomes activated when it recycles some of the peptides from the pathogen and present it on their MHC2 proteins. The helper T cell comes along and is activated by the macrophage. This helper T cell calls in reinforcements, including our killer T cell. The killer T cell's job is to monitor MHC1 proteins on the cells, on the surfaces of every cell of our body. And if it's normal, the killer T cell leaves it alone. It's normal, but if it's abnormal, what it's going to do is destroy the cell, which can also be a way to destroy cancer. Normal, normal, and this one is infected. So this bit of protein on the MHC1 protein, this bit of peptide on the MHC1 protein, is presented because this cell underneath here was infected. That would cause the killer T cell to destroy this cell before the virus or bacteria can replicate and cause even more infection. Now, the B cell, and that's cell-mediated immune response. The antibody-mediated immune response happens as the B cell is activated as it comes in contact with the pathogen and presents part of that pathogen. The helper T cell then comes in contact with the activated B cell, and we get plasma cells from that B cell, which will mass produce antibodies which neutralize and agglutinate the pathogen. That helps the eosinophils, which are similar to neutrophils, to engulf and destroy the remaining pathogen. This process is fairly short-lived, but we can have long-term immunity developed through the development of memory cells. These memory cells are the whole reason we get vaccines. So if we get a shot instead of a splinter, we are injecting ourselves with a dead or weakened version of a pathogen to go through this process, which takes time, so that we have memory cells backed up. Because if we ever get a second exposure to the same pathogen, these memory cells can immediately jump to mass producing antibodies to stop the spread of the disease without having to activate macrophages and helper T cells and B cells. So that is antibody-mediated immune response, cell-mediated immune response, and we began with a little bit of inflammation.